Geille from the European Federation of Patient Associations. And I will describe a little bit what is the Pharma Day. The Pharma Day is some kind of a meeting with all the community inside the room. We have the Pharma, we will present and report their activity in the domain of clinical trials or advanced studies in front of the patients. For the very first time, patients are behind the camera. They will have the meeting later on. And we started this program in 2021. For the first 3 years, we had 4 or 5 pharma inside the room reporting on their current work. And last year in Nijmegen, we switched to a face-to-face format. At this time, we had 5 pharma also in the room, but also 65 scientists and clinicians, because the meeting was attached to the IDMC 14 edition in Nijmegen in the Netherlands. And at this time, we made 2 decisions. The first one was to switch for the future in face-to-face mode. And the second time was to join with MDA for the further edition of the meeting. And so we had 65 people last year. And this year we are here. We had 5 pharma last year. We have 9 today. We will present in a few seconds their work. And I'm ready to announce that next year it will be connected to the IDMC 15 edition in Chicoutimi in Quebec. And that's it for us.
All right. Hi, everyone. I'm Tanya Stevenson. I'm the CEO at MDF. Very warm welcome to all of you who are new and who weren't here in the Cardiac Endpoint Workshop. We're so glad you could make it to Indianapolis. We are absolutely delighted to partner with Euro-DyMA, and we are grateful to Alain, to Bas, and to the entire Euro-DyMA family for inviting this unified approach for Pharma Day. I also want to just thank for a second all of you who had to travel from pretty far away, particularly those who are coming from outside of the country, because I know it is rather difficult time in this country right now for folks. And we really appreciate you making this a priority and making the myotonic dystrophy research community and our general community of patients and families your priority. So thank you for that. Also want to thank all of our sponsors, because we absolutely could not do this without them. So we have our lead sponsor for the conference, Avidity Biosciences. We have Dyne Therapeutics, PepGen, ARTHEx Biotech, Vertex, Sanofi, Novartis, AMO Pharma, Lupin Neurosciences. And today for Pharma Day, we also have Arrakis Therapeutics. As a reminder, just after Pharma Day, we hope that you will join us upstairs for the new early arrivals reception. And we are thrilled. We hope that you will really enjoy the ice cream bar, the mashed potato bar, and all the fun things that we will have for you there. So without further ado, I will pass this on to Andy.
Thank you, and welcome again also from me. So we want to try something a little different this year. Everyone knows about DMPK and the repeat, right? So we don't necessarily need to hear about the basics from all the respective presenters. Plus, we also have many, many more programs. So what we want to do as a first change, we have an official presentation from Nick Johnson, Dr. Johnson, from the NDM1 study, as it is a foundational piece, right, for a lot of the pharma and biotech programs. And then the next thing we want to do, we want to structure the blocks of presentations a little bit differently. So we have 4 presentations from the new kids on the block. So we give them 15 minutes and 5 minutes for question and answers. And then the last block, we hear from the established programs, and that's more like a speed dating approach. So 5 minutes or 6, not more. What's new? and then again also 4 to 5 minutes for question and answers. So I have the unthankful job to be the timekeeper. And I have set my phone to very loud. So please stay in your time slot so everyone gets to present and I don't have to stand on your toes. With this, I hand it over to Bas for the introductions of the speakers.
So as you can see, the collaboration between MDF and Euro-DyMA is quite okay. So it's now my turn to say a few words, really a few words. So I can introduce to you the program, the agenda, and I think I have to push this button. This is the agenda for today. And like Andy said, 2 parts, a part before the break and a part after the break. So easy is it? And yeah, the part 1 is done by the new kids on the block. Thank you for that word. And after the break, it's for the old faithful. They had already talks during our former Pharma days. So I give you the time to read the participants. One extra note, Georgina will not be able to join us today. Another remark is this meeting will be a recording and it will be available on the Zoom event platform for 30 days. And after that, we will make it public available, probably after some editing. And so you can have a look at it. So I hand it over back to Andy as the timekeeper. And first of all, Dr. Johnson will start. So Nick, I give the word to you.
All right, thanks for the introduction. You guys have actually heard enough from me already, but we'll try and make this brief. So really the goal is to give a high-level overview of some of the natural history data that's been collected and informed many of the clinical trials that our programs you'll hear about today. So these are my disclosures. So this is the current Myotonic Dystrophy Clinical Research Network. It has sites all over the United States, Europe, New Zealand, and Japan, and intends to expand more over the coming year. This is an update of the current studies. So at its foundation, there is a natural history cohort that is collecting patients at every age group. So the one that I'm going to talk about specifically today is the NDM1 study with the enrollment goal of 700, and we're currently just over 660 enrolled, so we'll be closing this year. Anybody who has myotonic dystrophy type 1 over 18 is eligible for the study, and the total observation period is 24 months. The Aspire DM1 study is just getting going, and that's really children with congenital childhood myotonic dystrophy under the age of 4, and that's 18-month observation period. And then beyond that, even newer is our treat extension, which is enrolling 150 children between 3 and 17 years of age with congenital childhood myotonic dystrophy. So in total, you can see those studies intend to basically capture any age of an individual living with myotonic dystrophy type 1. There are a number of sub-studies that's worth pointing out. So there's a longitudinal biopsy sub-study that seeks to capture longer range RNA-seq profiles. I'll show you some of the data from that. And then there's the COVID-19 sub-study, which is being led by Dr. Sampson, who I saw walk into the room, but there she is, which really start to kind of understand the impact of COVID-19. There's an actigraphy study that's being led by Tina Duong and Kate Eichinger, looking at activities outside of the study. There's an HHE sub-study that's being led by Mike Kiefer and Kate Eichinger, looking at ways to move away from the standard large fixed QMT system. And with that, I'll move into some of the current data cuts. So this is a study. Right now, this is a data cut looking at about 300 or so individuals, about 52% are female. You can see the mean age in the NDM1 study is about 44. The mean age of onset was about 25. One of the things that we've been trying to model and think about is how the repeat length captures in association with the age of onset. And so actually, if you start to plot the residual here, you can see that there is some variation, particularly at the edges of the repeat ranges. Looking at patient-reported reliability, the first graph here is a comparison between the short form of the MD high compared to the full-length MD high, and you can actually see that there's a very strong correlation between the short form and the long form, which is great to see as we move into shorter and more efficient studies. This is a graph here. Dr. Hamill was the first to really observe that we have been asking people their age of onset at every year. And so this is really just asking from baseline to 12 months, how accurate were those responses? And you can see that in general, if you use an ICC, the ICC is 0.99, but actually, for some of these individuals, they are highly variable. Many were quite the same between year over year. This is looking at the frequency distribution of the video hand opening time. So this is an important slide because really you can see here that, of course, we have some people here who have no myotonia whatsoever, but many people fall within this kind of 2 to 3 second range on the video hand opening time. And then, but of course, the same thing, there's many patients over here with quite long video hand opening times. This is a vector plot looking at the current change in 10-meter walk time from 0 to 12 months. The mean change is about 7.3%. I didn't mean to do that. There are some outliers here, but in general, you can see that if people start very fast, they may remain quite stable during that 12-month period. It's really in this group here where they've already had some disease progression where you start to see some of that change. Same idea, looking at hand grip strength here. Now looking at about 15.8%. This is a raw data cut, so you can see that there are still some outliers here that are likely driving that. But in general, if people are strong, same idea. They tend to remain quite strong over 12 months. For those individuals who had some degree of weakness, again, that's really what's driving that change over time. Just to harken back to the last session, this is one graph here that Sam Carroll was looking at. Here you can see this is the PR interval plotted by age, by years since age of onset, and by repeat length as measured by clinical samples. And so you can see really no relationship with any one of those. In another study looking at using MD StarNet with 429 individuals here, again, you can see that there's no relationship between the degree of risk for cardiac dysfunction as well as the age of onset or repeat length. One of the real challenges that we've had in the network is trying to understand, how to capture the cognitive profile that we know is associated with the disease. We sought to do this using this measurement called the CogState, which is a computer-based assessment tool. We chose that because it's available in multiple languages. You do not need a neuropsychologist to administer it. You just put the computer in front of the patient, and it kind of captures cognitive function. So the first thing that you might notice here, or maybe to point out here, is that if you look at the global set of participants, that on average, many of these outcome measures are pretty, so these are Z-scores here, and the directionality indicates whether or not it's better or worse on that outcome measure. And so you can see that for many of these cognitive functions in the larger cohort, that they actually score pretty normally. But the other thing to point out is that same concept is that in many instances, these are not normal distributions. There is skew in the population that you wouldn't normally expect to see on a Z-score distribution. And so we did a latent factor analysis and really identified 3 different groups of individuals. So in about 60 or so individuals in that cohort, they had this kind of below average score. And the test items that are in red are what's below the average value. And these is my description of what these tests are actually telling you, which is difficulty with psychomotor function, impulsivity, executive function, memory, attention, and verbal learning, which is what we would expect to see. But equally striking within the cohort was that there was about 60 or so individuals with pretty normal scores on those same outcome measures. And there was another 120 individuals that actually were a little bit above average. So I think, this is really a lesson for us that we need to continue to work harder to identify better cognitive measures that stress the population, but also, many of these individuals that are willing to drive to a clinical center and have the ability to do a natural history study are maybe not the ones that are most severely impacted from a cognitive standpoint. And then the last thing I'll touch on briefly is this RNA splicing index, which was really initially developed by Charles Sorton, who I also saw in the room someplace, and then has been published recently by Dr. Hale and Maria Provenzano. And so this is just looking at here in this cohort, a correlation between the splice index, which is a 22 item splicing events, compared to the a variety of different clinical measures. So this is ankle dorsiflexion, this is hand grip strength, and this is a 10-meter walk run. And you can see that there's really quite strong correlations between the baseline value of the splice index as well as the baseline value of the outcome measure itself. Moreover, what was a little bit surprising actually is that when we started to look at the baseline splice index versus the 3-month outcome measure and 3-month splice index, you can actually see that if you fit a model looking at the baseline splice index and ankle dorsiflexion, they're actually quite good predictors of where the physical function will be in about 3 months' time, suggesting that there may be some predictive value for these individuals as well. And with that, I definitely am kept to my time. I didn't hear the buzzer go off. I don't know if are we taking questions or are we just moving? Okay, all right. So this is really a big thanks to our funders, which you can see here, and a special thanks and kudos to our investigators. I tried to do outline of Larry and Carlene, and then Man Hung, who provided some of these data. Okay, now I'm done. Thanks.
Thank you very much. Nice talk. Matteo Garibaldi from Rome. It's very interesting that the splice index could be a surrogate biomarker that can be used as outcome measure. But do you think that it could be used in clinical trials or clinical practice to monitor the disease progression and the response to treatment?
Yes, it's a good question. So certainly out of the biomarkers we have, I think composite measurement of RNA splicing appears to be the most attractive biomarker that you choose at this current moment in time. It's nice that it may have a little bit of predictive ability. I kind of glanced over it, but really to this point, we've only measured RNA splicing in the natural history study out to about 3 months. And so we're working on collecting biopsies to see about the long-term outcome. So I think more work will go into this to really kind of tell us how good it is in terms of long-term predictions of individuals. From able to do this in clinic, that's a great question as well. We're working on a process where you would be able to, input the data on a computer interface, and it would spit it back out, because I didn't go through it today, but it's certainly in the paper that there's a reference library that normalizes the individual's splicing index to a larger population. And so that's really kind of the trick to get that into the clinic. So not quite yet. All right, Andy, go ahead.
So I think next we will hear from Modalis Therapeutics, and we have here Dr. Connolly. He's the director of molecular biology and genomics. Take it away.
Yes. Well, thank you. All right. Thanks for that introduction. And thanks to the MDF organizers for giving Modalis a chance to talk about some of our work here today. Yes, we're the new kids on the block, but I'm looking forward to introducing ourselves at this meeting and meeting some folks over the weekend here. Modalis has been developing a CRISPR-based platform for gene modulation. It's composed of 2 fused components, a Cas9 portion that can target this platform to a specific portion of the genome, and then a transcription modulator that can affect gene expression once it's there. The application of this platform is to activate compensatory gene expression or knock down toxic gene expression as a treatment for genetic diseases. As I'll discuss today, we've been working on delivery strategies to express this in therapeutically relevant tissues and cell types. And our hope is that we can use CRISPR interference as a treatment for DM1 by knocking down DMPK mRNA expression. This audience doesn't need an introduction to DM1, but I'll highlight that in DM1 patients, DMPK mRNA can harbor extended repetitive region at the end of the DMPK mRNA transcript. This repetitive region is thought to cause nuclear aggregates, which can sequester splicing factors and alter gene expression. These changes in gene expression can result in some of the clinical phenotypes that we see present in DM1 patients. Modalis is proposing to reduce these transcripts through transcription inhibition of using CRISPR-I to rescue some of these phenotypes. Again, we're newcomers, so new kids on the block, but and there's to this field, and there's a lot of folks that have been working on a number of other therapies in this field, including small molecules that might be able to sort of break apart these aggregates and prevent sequestration of protein factors. Another has been oligonucleotide-based therapies, which can target the mRNA for target DMPK mRNA for degradation. One challenge here has been delivery, but there's been some interesting work recently using antibody conjugates to solve some of these delivery problems. I think we'll hear more about that after the break today. Our approach has been a gene therapy approach where we're looking to package the platform that I described a few slides ago and deliver specifically to muscle cells and cell types for knockdown of DMPK expression. So for the remainder of this talk, I'm going to talk about some of the work that we've been doing to set this up as a therapeutic. I'll talk about designing guide RNAs that are potent and specific for DMPK knockdown, some of the in vitro assays that we performed using patient-derived cells. And then I'll talk about packaging this CRISPR gundam into AAV particles for specific delivery to muscle cells. Finally, I'll end showing some experiments in where we were able to knock down DMPK in DM1 mouse model. Okay, so we began this project by designing CRISPR gundam compatible guide RNAs around the promoter region of DMPK. We try to be fairly comprehensive in the guide RNA design here, but we do try to keep the guides so that they're compatible with multiple genomes, including mouse, non-human primate, and human. And we do this because we'd like to develop one construct or one therapy and use that for preclinical studies that we can pass to the clinic from following the preclinical work. To test these guides for suppression of DMPK, we packaged the CRISPR into lentiviral particles and transduced immortalized human myoblasts. We measured the DMPK suppression using qPCR and plotted on this chart here. We tried to look for guide RNAs that could induce DMPK suppression by 75 to 80%. And it's no surprise that a lot of these guides are located around the DMPK promoter region. But what's interesting is that some of the guides didn't work very well, even at the DMPK promoter region. And I think that this emphasizes the importance of testing these guides in cell models. We took 3 of our top guides and wanted to compare them to the control guides. So we used next generation RNA sequencing to show reduced transcript levels. Here we did see some effect on neighboring genes, but we thought that these effects were fairly minimal compared to the almost complete knockdown of DMPK transcripts from the RNA-seq data. For these 3 selected guides, we also looked at global effects of gene expression using volcano plots that compared the targeting guide to the control guide. For each of these, we saw significant knockdown of DMPK. However, there was some variability in the off-target profiles for these targeting guides. We chose to move forward with guide RNAs 2 and 3, which seemed to show more specific knockdown of DMPK, and avoid guide RNA 1, which seems to show some more promiscuous gene expression changes. Okay, so next we wanted to ask if inhibiting DMPK transcription can rescue some of the molecular phenotypes associated with DMPK, with DM1. On the next few slides, I'll show some experiments that test the reduction of nuclear DMPK transcripts as well as splicing abnormalities. To test for nuclear accumulation of toxic mRNA transcripts, we used immortalized myoblasts derived from DM1 patients. As expected, these cells had a higher tendency to show nuclear RNA foci corresponding to CUG repeats. When these cells were treated with Gundam and selected guide RNAs, we saw up to 85% rescue in cells with nuclear CUG foci compared to control cells. These results suggest that inhibition of DMPK transcription can rescue some nuclear accumulation of these transcripts. Next, we wanted to test for rescue of aberrant splicing phenotypes in patient-derived cells. For this, we use PCR primers that can identify mRNA isoforms associated with DM1 disease. As an example for this assay, PCR primers PCR products are resolved that distinguish TNNT2 isoforms that lack exon 5 and are more abundant in control cells from isoforms that include exon 5 and are predominant in patient-derived cells. By measuring the band intensity in these gels and finding the ratio of these two mRNA isoforms, we can quantify the splicing differences in these cells. And for TNNT2, we found that treatment of Gundam and candidate guide RNAs shows rescue of these splicing phenotypes, restoring splicing profiles closer to that of control cells. We also measured other transcripts that have been reported to be misspliced in the literature and found that treatment of Gundam and candidate guide RNAs shows varying levels of splicing rescue. So with this program and experience with other programs at Modalis, we set out to design an optimized system for delivery of CRISPR Gundam to target tissues. When thinking about the desired delivery and expression of Gundam, we consider potential serotypes, AAV serotypes, as well as the sequence characteristics of the AAV genomes that can be optimized for expression. On the next few slides, I'll discuss the packaging of these genomes, but first I'll present some studies investigating potential AAV serotypes that can be used. The gene therapy field has been growing with the focus on improving natural serotypes for better transduction in target tissues and cell types, which is particularly true of muscle cells. Here we tested transduction of some muscle-tropic serotypes and compared with the natural AAV9 serotype. We found that muscle-tropic serotypes were capable of better transduction in the heart, diaphragm, and TA muscle tissues compared to AAV9. But we also found that the muscle-tropic AAVs had some liver detargeting properties, which could lessen some of the concerns of liver toxicity that's often associated with gene therapy. These studies have allowed us to improve targeting of Gundam to the right tissue and avoiding unwanted side effects. Okay, so next we wanted to analyze the sequences in the AAV genome. We worked to identify potential sequence elements that help to maximize platform expression in the target cells and keep the genome small enough to fit into an AAV vector. Some of the elements that we've tested are promoters, poly-A signals, and some coding variants for the for the CRISPR-I transgenes. We also need to keep in mind that some of these elements might have sequence complexity that isn't always compatible with AAV packaging. So in the next few slides, we set up some experiments to test packaging of the complete AAV genomes. We set up protocols using long-read sequencing to sequence the entire length of the AAV genomes from our AAV preps. And these protocols let us assess the coverage of the complete genome to get an idea of any potential problematic sequence elements if they exist. This is an example of pretty well-behaving AAV where we saw complete coverage of the AAV sequence on the top line here. When we looked at sequencing at the ends of the sequencing reads, we found that the 5' ends associated with tag mutation sites for our sequencing prep were pretty uniform throughout the body of the AAV. This was expected. And we also found that sequencing reads at the 3' ends were largely at the anticipated end of the AAV genome. This, together with some gel analyses, led us to conclude that this was a pretty well-formed AAV and that this AAV genome was completely packaged. On the other hand, we also had genomes that were less compatible with packaging, like this one, where we found coverage of the genome was more staggered, and there was multiple sequencing ends throughout the AAV genome that were unexpected. However, these positions at the sequencing let us identify smaller package genomes by pulling out the sequencing reads that were associated with these peaks throughout the body of the genome. I'll mention that these AAV constructs produced preps with lower titer, and that we were able to detect these truncated genomes in agarose gels as well. Taken together, though, these experiments let us identify this promoter as a problematic sequence that should probably not be included in a final product. Okay, so after some quality control experiments showing that we can expect a fully packaged genome, we tested these AAVs in vitro using human myoblasts and cynoskeletal muscle cells. Here we were able to show that with 2 test articles, we could get significant knockdown of DMPK in vitro. Oh, sorry. Yeah, this gave us some confidence to move forward with in vivo experiments. And for these experiments, we use the DMXXL mouse model that needs no introduction here, I'm sure. But this is transgenic and carries a human copy of DMPK with more than 1,000 repeats in the 3' UTR of DMPK. Unfortunately, we weren't able to observe strong phenotypes with this mouse, but it was a model where we could test for DMPK suppression using our CRISPR-I platform. For this expression, adult mice were systemically injected and muscle tissues were harvested after 4 weeks of the in-life experiment. And we found a statistically significant knockdown of DMPK in the majority of the samples tested. Interestingly, we saw a strong knockdown in heart muscle, a critical target organ for DM1 patients. We also saw a significant DMPK reduction in skeletal muscles, including the diaphragm, using one of the test articles. These data suggest that these test articles can be used to alleviate some of the molecular phenotypes as well, and that's what we tested on the next slide here. We use fish experiments to measure accumulation of repeat-containing mRNA in the nuclei of the heart tissue, and here we see a reduction in the accumulation of nuclear foci corresponding to DMPK-CUG repeat mRNA. These data suggest that molecular phenotypes can also be reversed by treatment of CRISPR or a CRISPR-I platform in vivo. Okay, so I'll end there and I'll say thank you for your attention while I describe some of the work that we've done to develop a therapeutic for DM1 using CRISPR-I, and through potent and specific guide RNA design, some in vitro test, packaging of this platform in AAV, and then some in vivo some in vivo experiments in a DM1 mouse model. Finally, thanks to all the dedicated folks at Modalis. Here they are working away at our site in Waltham, Massachusetts, just a few miles west of Boston. And thanks again to the MDF committee for putting on this great conference and the invitation to speak.
Yeah, here.
Yeah.
So how much off-target do you see for your guide RNA?
Coming in.
How much off-targets you see for your guide RNA? And
so for the guide RNA, the ones that we moved forward were, we showed the volcano plots. So there were very few significant off-targets for the 2 that we moved forward with. There was one of them that showed very high off-target.
Okay.
Yeah.
And the DMPK you measure here, the toxic DMPK or the total DMPK?
In the mouse model, or
Yep.
In the mouse model, no, that's the human DMPK transcript, the transgenic mouse. So the mouse also has a mouse DMPK version.
Hi, thank you. I was wondering, so you package your Cas9 and your guide all together inside one AAV particle.
Yeah, that's right.
Just quick question: which Cas9 do you use? Is
This is SA Cas9.
The SA Cas9. Yeah. Okay, that makes sense. Thank you.
Hi, thanks for a nice talk. I'm wondering how efficient is your CRISPR in terms of like the % of cells that are modulated with your CRISPR strategy?
Yeah, so with in the mouse, it's pretty easy. The IHC shows probably 60 to 80% coverage transduction. Yeah.
How durable is the expression in monkeys?
So in monkeys, this one hasn't been tested in monkeys, but we have another program for Llama 2 CMD This is showing good durability in monkeys. And there's a paper on bioRxiv right now that you can have a look at. But in this paper, we were able to see good transduction and durability in non-human primates.
Thank you for your presentation. Very nice. It seems to me that in the mouse model, the DMPK in diaphragm was not very significant, and one and the other was very modest. Any explanation?
In the diaphragm, I'm not completely sure. I mean, I think a lot of this has to do with the systemic delivery, which sort of goes right to the heart. So I think that's why we see the most profound suppression in the heart, and then we kind of hope that it passes through. So I think a lot of that has to do with the efficiency of delivery to the different tissues. Yeah. Great. Well, thanks for your attention. I appreciate it.
So next, we'll have the Sanofi team here, I think, with Dr. Galeon and Dr. Goulet.
Great. It's a privilege to be here and share with you some of the innovation that is happening within the genomic medicine unit at Sanofi. These are disclosure before we proceed. We're all employees of Sanofi when this work was conducted. Sorry, moving forward. So what Genomic Medicine Unit, the GMU for short, at Sanofi, we are developing technologies to address several diseases with high unmet need. We have a very dynamic portfolio spanning non-viral delivery and viral delivery that encompass several technologies. And just to give you a small brief example here of all the different work we're working on, we have vectorized antibodies, and there's a lot of work happening in this field. We have newer technologies that we will present at the upcoming American Society for Cell and Gene Therapy. We have extensive work done with RNAi to silence abnormal disease-causing transcripts, and I'll show you an example today. We have already in the clinic with protein replacement therapies, so that's very exciting. And we're pushing the boundary now. We're something quite unique. We're doing in vivo cell reprogramming and gene editing. So that's a very exquisite and beautiful technology. So we look forward to see what these technology can bring to the patients. More in focus of the topic today about DM1. I won't go over the disease and the molecular biology behind the disease. Just want to highlight here on this slides the CUG repeats in the transcript, and that's the cause of the disease. That results in RNA foci and splicing aberration. Our technology is really designed to go to address the root cause of DM1 and go after the DMPK abnormal transcripts. There are four main features of our technology. The first feature is we have a very specific artificial microRNA that will target DMPK. The second feature is we're using a myotropic capsid we call AAV711 that will deliver a high level of the genome to the muscle cells. The third feature is we are also using a myotropic promoter, so the expression is very high in the targeted cells. And this is meant to be a one-time administration for long-term duration, steady state level of the microRNA for the M1 patient. For the first feature, I was very enthusiastic when I put this slide together. There's quite a bit of information in there. Let's see if I can point. We have two models of DM1, human patient mortalized DM1 cells with large number of CTG repeats. And we have the DMXL mouse model that was mentioned previously, very widely used. In both models, we have a very nice suppression of the DMPK transcript here in vitro and also in vivo in the mouse, several of the skeletal muscles and the heart. We have a reduction, beautiful reduction of the RNA foci in vitro and in vivo. And change in splicing events were measured here. Showing just an example of the in vitro data. We have complete RNA-seq data, both in vitro and in vivo. You can come to our posters and take a look at that data. Thousands of events were measured. The most exciting part, I think, for this slide is at the bottom here on the left, where we have a deficit in cardiac output in the untreated DMXL mouse model that is restored with the treatment with our microRNA. So that's it's a very elegant data that our microRNA is doing something in the heart. The other important piece of data here is the myotonia. It's hard for me to point, it's so far. But these DMXL mouse model, these animals are very mild myotonia. It's actually hard to quantify, but it's completely absent in wild type animal. It's present in a large fraction of the DMXL mouse untreated animals. And there are much smaller number of myotonic events that can be detected following our microRNA treatment. So we are very excited with this data. The second feature of our technology is a myotropic capsid. We have done multiple experiments in non-human primate. We showing an example, just an example here of GFP expression under a ubiquitous promoter. We see much higher expression in relevant skeletal muscle, tibialis anterior, quadricep, gastrocnemius, bicep, tricep, diaphragm, heart, with our capsid. In a different experiment, we've done that multiple times. We also performed the immunohistochemistry, and we can see here the signal is much higher to a reference capsid. Here we use for comparison is AVRH74, which is a capsid that is currently used in the clinic. So we have a very potent myotropic capsid. Now, if we combine the microRNA with the myotropic capsid, this time expressed under a myotropic promoter, what you can see, well, my apology. What you can see on the graph on the left here is the amount of DMPK suppression in several skeletal muscle. We've tested at low, medium, and high dose. There's a very nice dose-dependent reduction of DMPK transcript in all of these skeletal muscles. It's a fantastic data because we reach up to 90% suppression, silencing of DMPK transcript in these animals. If we go to look at the graph on the right, this is the microRNA expression level in these tissues. We have very high expression. It's dose dependent in all of these tissues. So higher expression in muscle cells. There is slight expression in the liver, in the dorsal root ganglia, but it's not associated with a significant reduction in DMPK transcript. So this attribute makes for safer therapy moving forward. We have also collected images looking at the distribution of the microRNA and the silencing of DMPK. So this data strongly support the PCR, digital PCR data just shown on the previous slide. The image here on the top left shows the microRNA expression here is in green throughout the heart, a very widespread distribution, high level. A little bit more difficult to see, but also widespread expression in the tibialis anterior. And when we look at untreated animals, there's no microRNA, as you would expect. This is the staining for the DMPK transcript in these animals in red here throughout the tissue section is almost completely absent following the SAR 446268 treatment. And same thing in the tibialis anterior. It's a smaller, lighter amount of DMPK here endogenously in the TA, but it's also completely gone following the SAR 446268 treatment. We have performed a very comprehensive IND-enabling GLP tox study in juvenile cynomolgus monkey. And very happy to share that there's no test article related finding observed in this study. We did see minimal to mild change in coagulation parameter that resorbed. They were transient. We also see transient elevation of liver enzyme, mild to moderate level of ALT and AST, which are a little bit expected from an intravenous AAV dosing to target skeletal muscle. The typical biodistribution and shedding pattern were observed, and we measured the vector genome and all the relevant tissue for this disease. So we're very pleased with that. the no adverse event level established at the highest dose tested, which is 6 times the safety margin from the starting human dose. So this is a very safe therapy moving forward. I'll conclude here, and then I'll pass to my colleague, which will tell you a little bit about our clinical plan. So in summary, we created an innovative delivery platform with a myotropic AAV, a myotropic promoter, and a very potent microRNA. We show a target engagement in the human DM1 human cells, and we have a good proof of concept. We have a good proof of concept in the animal model, the DMXL mouse model, showing a reduction of DMPK, a reduction of the RNA foci, change in the aberrant splicing. Most important, reduce myotonia and improve cardiac function. In non-human primates, we see a dose-dependent distribution, target engagement, and all those tested were very well tolerated. So this makes our 446268 has the potential to deliver a one-time disease-modifying therapy that may confer life-changing long-term benefits to a range of DM1 patients, and that's supported by a good safety profile. On the next slides, before we transition to the clinical plan, just want to acknowledge all the effort and know-how from several scientists within the genomic medicine unit at Sanofi, but also the larger Sanofi ecosystem that permitted to bring this program to this pivotal stage. So this is forward, backward.
Good afternoon, everyone. Thank you for the opportunity to finally present to you for the very first time our BRAVE study. So my name is Belle Galeon. I am the Global Clinical Lead for the DM1 Gene Therapy Program at Sanofi. And this is our study, the BRAVE study, is the first in human phase 1 and 2, single arm, open label, multi-center global study. So as you can see here, this study consists of 2 parts. We have the part A, which is the dose escalating part of the study, as well as part B, which will be the dose where we will select the dose to expand the study. So part A, we will enroll adults ages 18 to 50 years old. As you can see, we will test 3 doses with 3 participants per dose cohort and the data review in between doses. So once we have dosed all the 3 cohorts, in part A, we will choose a particular dose that exhibits safety and efficacy, and we will move on to part B, wherein we will include a pediatric population. So this population will be participants aged 10 to 17 years old. All participants in this study will then roll over to a long-term follow-up study. So 2 years in our main study, 3 years in the long-term follow-up for a total of 5 years. So this is our eligibility criteria. So this study is limited to non-congenital DM1 patients, ages 18 to 50 for part A, 10 to 50 for part B. Patients with confirmed DM1 based on CTG repeats of at least 50. Patients need to be ambulatory with or without assistance, in reasonably good health and clinically stable. There should be no significant neurocognitive issues. So in this study for part A, we will include only participants with moderate cardiac risk and implanted pacemakers or ICDs. However, in part B, we will consider participants with perhaps low and some higher risk cardiac risk participants. So exclusion, we will not include participants who have ejection fractions of less than 50, who have seropositive, are seropositive to the SAN11 capsid, with active malignancies, with contraindications to corticosteroids, since we will be using steroids in the study, and those who have active infections. In terms of primary endpoints, this is a safety study, and we will be looking at treatment emergent adverse events. Additionally, we will be looking at efficacy in Part B, looking at least 40% DMPK mRNA knockdown in the muscle biopsy. Additionally, we will be looking at functional measures, including 10-meter walk-run test, change in myotonia, as seen by your hand opening time, the hand grip test, DMPK mRNA level changes, and changes in the RNA splicing index, as well as AAV vector shedding. Additionally, in Part A, we will also be looking at the proportion of participants with at least 40% DMPK knockdown. So this study is available on clinicaltrials.gov, and we should be recruiting shortly. So for more information, please visit clinicaltrials.gov, and we do have posters that we will be presenting tomorrow and on Saturday. Thank you so much for your time and attention.
It is a new capsid designed by Rational Design.
Hello, nice talk. So my first question is, you have shown that this AR446268 showed significant promises in improving cardiac function. So what was the concentration of that AR446268 after the treatment? And my second question is, what challenges your team has faces during this AR44 treatment in human?
I did not get the second question. What's
So,
the second question?
Yeah, what challenges your team has found during this AR446268 treatment?
I can start with the second question. So, to clarify, the study is still it is on clinicaltrials.gov, but it's not currently recruiting, and so there have been no patients have been infused yet. In terms of challenges, then I guess as we saw here, the capsid seems to be fairly safe. We have a good safety profile, and so our particular challenge, I think, in developing is making sure that we are able to do a good cardiac surveillance in our protocol, and ensure that we have safety measures accounted for in this population with a very heterogeneous presentation.
And a quick follow-up question. You have shown that you have excluded hepatitis B and C patient. So why you have chosen this exclusion for hepatitis B and C patient? Yeah. In a patient choices for the human study, phases, how you, what are the criteria for the patient in inclusion and exclusion?
I'm not entirely sure I understand the question, but I think it had something to do with the cardiac risk. Okay. Thank you. So as we know, there is good transduction of the capsid in the liver, and so we're also aware that a lot of DM1 patients already have some abnormalities in their liver profile, and so we wanted to make sure again that patients are safe, and so we want to exclude participants with perhaps pre-existing liver disease, as well as the fact that we will be using steroids, and so there is immunosuppression, and so we don't want to activate any latent and other infections.
And for the first question, I can say that we are not revealing the dose that we're using in the non-human primate, but we have a very nice understanding of the relationship between the amount of microRNA and the DMPK suppression that translate across in vitro, in the rodent experiment, and the non-human primates. So we have a very good understanding of that.
Thank you. So two brief questions for the non-expert. Could you briefly comment what the rationale to select the artificial microRNA versus other approaches? And what would be the mechanism of action to target and to inhibit, reduce the DMPK? And second, what is the durability of the result of the transduction? Do you have data longitudinal that is maintenance of the transduction at months, 1 year?
Yeah, the expectations are that this therapy will be expressing the microRNA for several years. We don't know the extent of that duration. It'll probably be more than 10 years that we will need to assess it. The selection of microRNA, it's a steady state, constant level of the silencing moiety that will target DMPK. So there's no need for reinfusion or anything like that. It's always there, it's always on, it's all the time. So it should be very consistent without trough and valley. So that's one of the main reasons we choose this technology.
Thank you for the work. I think in your presentation, you showed the improved cardiac function, but if I remember, that was in a skeletal muscle specific mouse model, was it? So have you tested this in a heart-specific mouse model of DM1 to see if there's any change?
We haven't yet tested that in a heart-specific mouse model. No, that hasn't been done.
Thank you.
What was the reason to not include the CDM? So it's only non-congenital forms of DM?
So I think Martin alluded to the fact that we did not get good transduction in the CNS. And so as we know, a lot of congenital patients present with neurocognitive, significant neurocognitive issues. And so at this point, we feel that, the therapy would best be most suitable to participants who do not have or do not present with that severe neurocognitive issues. However, we do anticipate that with improvements in your neuromuscular function, there may be improvement in fatigue, and so there may be also some indirect effects from that.
I was wondering, is the microRNA specific to the expanded allele, or will it also inhibit the other copy? And the second question is, you've used the microRNA, which increases the possibility of off-target effects because microRNAs do not need perfect complementarity to inhibit other genes. So any thoughts on that? So for the first question, hold on, I lost my train of thought. The first question is, no, it's not allele specific. It will suppress, silence total DMPK, both the mutated and normal transcript. We know it's safe from rodent study where knockout animals are perfectly fine. And we know also from our own work where we see up to 90% suppression now, and there's no histology, no finding, nothing at all. So we believe this is safe. Regarding the, oh gosh, sorry, I'm blanking again. The off-target, we have done extensive work for that. We did found few off-target, but we don't consider them to be of any concern at all. So as expected with microRNA, siRNA, or oligonucleotide, you will always have some off-targets, but what we see is a very clean profile.
Thanks very much for the great presentation. Given that there is expected to be long durability with AAV delivery here, is there a level of DMPK knockdown that might be too low here? And, because you're targeting that 40% knockdown, but your preclinical studies show you could get upwards of 90%. So have you guys thought about that?
So that's not our goal. Our goal is not to obliterate and completely eliminate the expression, but to alleviate the load on the system and have less of those RNA foci will be key, I think, for this disease. And that's really the goal. I think it's very safe. It's likely that this protein has other redundant protein that fulfills similar roles.
So next, it's pretty exciting, right? We hear about these new delivery methods, and now we have actually novel therapeutic forms, which we hear from Dr. McVie from Vertex Pharmaceuticals.
Thank you. Hi, so good afternoon, everyone. First of all, I'd really like to thank MDF and Euro-DyMA for the invitation to speak today. As mentioned, this is the first time that we've spoken at Pharma Day, and we're super excited to be here. I hope that you don't mind, but I'm going to spend a couple of slides just to actually introduce Vertex, given that this is the first time that we spoke at this meeting. So with the next slide. So Vertex is a global biotechnology company. You can see here that we invest in scientific innovation to create transformative medicines. So we're not in the business of me too medicines or medicines that may have incremental, that may have incremental benefit, but we really aim to develop medicines that are truly transformative for patients that live with serious diseases. And from a commercial point of view, that then allows us to really focus on specialty markets. We work on really tough diseases, and we do that because at the end of the day, patients are really at the heart of what we do. We aim to strike at the core of those diseases, i.e. we develop treatments that target the underlying root cause of the disease. And that's a key part of what we do in our R&D strategy, is to understand that underlying cause of the diseases that we work on. And we're not afraid to take on the impossible. So our overall corporate strategy, as well as the business model that we have, is really a blueprint for serial innovation. So from a research and development point of view, we really, as mentioned, we combine transformative advances in the understanding of human disease, and then we can pair that up with the innovative science to develop a therapeutic that is really designed specifically to target the root cause of that disease. So with that, we focus on targets that are validated by human biology. We then spend a lot of time creating predictive lab assays and clinical biomarkers so that we can really move quickly through development and identify that rapid path to get us to registration and approval. And as mentioned before, we really work on therapies where we believe we can have that transformative benefit. And we do that regardless of modality. So we basically identify what the problem is within the diseases that we work on. And then we figure out what is the right tool to solve that program. We have multiple disease areas and multiple programs at Vertex where we use multiple tools and different modalities to treat the same disease, whether it's for a different subpopulation of the disease, whether it's for patients with particular mutations in the disease. But by really understanding the human biology, we can really discover and develop the right therapeutic for those patients. And with that, as you can tell, we really like to put science first. And we really believe that that's how we're going to unlock value, not just for us as a company, but most importantly for patients that live with serious diseases. We invest the majority of our OPEX in research and development, and as such, despite being a company that has multiple marketed products, actually 3 out of 5 employees at Vertex actually work in research and development. And that really allows us to then have that serial innovation platform where we really don't stop with the first molecule, but we continue to raise the bar until we have a treatment for patients living with diseases that really gives the maximum benefit and potentially aiming to develop a cure. Through that serial innovation and with that corporate and R&D strategy, we really have a broad pipeline in multiple complex disease areas. You can see here, many of you in the room may know us from our cystic fibrosis portfolio, but we also have molecules that we're developing and now have on the market for genetic kidney diseases, for hemoglobinopathies like sickle cell and beta thalassemia. Pain, trying to transform how pain is treated through some of our molecules that are in the market and being developed there, as well as type 1 diabetes, and importantly for this group, our muscular dystrophies disease area, where we work on Duchenne muscular dystrophy as well as myotonic dystrophy. All right, so with that, I am excited to be able to introduce our investigational molecule that we are evaluating for the treatment of DM1. And this is a peptide conjugated oligonucleotide. I do not need to describe this slide for the people in this room in terms of what the underlying biology is in myotonic dystrophy type 1. But suffice to say, because there's been so much work done here to understand the molecular basis of this disease, we can really then design potential therapies that really aim to address the root cause of the disease here. And with that, we believe that our approach that we're looking at is that to target the toxic hairpin using a selective steric blocking oligonucleotide that is targeted to the mutant RNA and the RNA hairpin to sterically block the binding and prevent the sequestration of muscle-blind and other splicing factors to that RNA. Back in December of 2022, we announced that we had in-licensed a molecule from Entrada Therapeutics. That molecule is now named VX-670. This is our investigational peptide conjugated oligonucleotide. As you can see, it contains that active oligonucleotide component, which is a PMO, which is a type of oligonucleotide that can function in that steric blocker mechanism to the toxic RNA hairpin. But of course, that oligonucleotide has to get into the cells, and we do that by conjugating it to peptide moieties that allow delivery into the cell and, more importantly, into the nucleus of the cell. So how does that work? This cyclic peptide is designed to bind to the cell membrane. It's then internalized into endosomes, bringing the oligonucleotide with it. The cyclic peptide can then induce endosomal escape, so the oligonucleotide can then get out into the cytoplasm of the cell. We then have another peptide that's a linear peptide that's designed to be a nuclear localization signal that can then take the oligonucleotide into the nucleus, which is the site of action and where the mutant or the toxic RNA molecule is. So you can see from a delivery side of view, we've really thought carefully about all of the barriers that that active oligonucleotide needs to get through when it's administered into the circulation to get it into the nucleus of the cell. Once it's inside the nucleus of the cell, as mentioned, the oligonucleotide can then bind to the RNA hairpin. It can release the muscle blind with the potential of correcting splicing. Our preclinical data suggests that there is a role for this and the potential to be beneficial and work as designed. You can see here that in preclinical models of DM1, including patient cells and DM1 mouse model, you can see that we can reduce RNA foci in human cell models of the disease. We can also correct missplicing following treatment. We can do that in multiple human cell lines, as well as in the mouse model of the disease. And in the mouse model, we use the HSA-LR mouse model, so we can also evaluate myotonia and show that following treatment, we can resolve myotonia in that DM1 mouse model. So with that, and with the very encouraging preclinical data that we've generated, as well as a nice safety data set, we're happy to communicate that we have moved this into clinical trials. So our clinical trial is called the Galileo study. And so this is a phase 1/2 study. It's placebo-controlled. You can see it's in 2 parts, part A and part B. Part A is that single ascending dose portion of the trial, and part B is a multiple ascending dose portion of the trial. It's in adult patients at the moment, non-congenital patients that are genetically confirmed with DM1. We will evaluate primarily safety and tolerability in this study, but also look very closely at the pharmacokinetics as well as the pharmacodynamics. And the pharmacodynamic measure that we're most interested in is the correction of splicing. And we'll do that through obtaining muscle biopsies. And Galileo is a global study. So we now have sites active and recruiting in multiple continents. You can see here, we have sites in the US and Canada, in Europe, the UK, as well as Australia. So with that, I would like to say a huge thank you first to the patients who have been contributing in our clinical trial and hopefully will continue to be excited about our program. 2, to our trial investigators and the clinical sites that are helping us with this study and moving along quickly. And 3, of course, to the advocacy organizations who have brought us here today and worked tirelessly to support this community. And I'd also really like to thank the Vertex Muscular Dystrophies cross-functional team. So thank you. With that, happy to take questions.
So does your technology target multiple CUG repeats? There are a lot of normal transcript in the human transcriptome that have more than 6 CUG repeats. So will those be affected by the technology?
So we don't believe so. So we did that. So in some respects, that's a little bit of the off-target work that we've done, is to look, as you say, other CUG repeats across the genome. And in our analysis of that, we do not see an effect of binding those CUG repeats. So we do believe that this is specific to the repeat expansion in DM1.
I noticed your time point is 15 days post-dosing. I'm interested in hearing more about the rationale behind that.
Yeah, I mean, that's really coming from our preclinical data package, and that we know that following a single dose, we see splicing correction very quickly. And therefore, we knew that we can look at muscle biopsies very early in the clinical trial. And we believe that that time point is when we potentially will see the maximum splicing correction.
Thank you. Maybe I've missed it, but you have data regarding the biodistribution as part of the skeletal muscle. And what about the myocardium, heart, and other tissue delivery?
We do. We didn't present that today. But yeah, so the cyclic peptide delivery approach is not cell type or tissue type specific. So we actually see really nice broad distribution of our molecule in our preclinical studies, including to the heart. As well as skeletal muscle, as well as in smooth muscle. One thing I will mention is that we don't think it crosses the blood-brain barrier very effectively. So we do think that that potentially is one limitation of this molecule. And it's something that we're working on for next generation molecules.
Thank you for your presentation. Very nice. Can you give us more details about the administration and how often is administration and multiple ascending dose?
Yeah, so in the multiple ascending dose portion of the trial, it's dosed every 6 weeks. And that portion of the trial is 3 doses, so over a 12-week period.
About the administration?
IV infusion. Infusion, around 30 minutes. 30 minutes. Yeah.
So closing the loop, right? We go from new delivery to maybe more broadly bioavailable inhibitors to now following the SMA example, right? The first generation, hopefully, of orally available drug candidates. And we welcome here from Arrakis, Dr. Mahoney. She will tell us about her results. Thank you.
Great. Thank you to the organizers for inviting us to come give this talk. My name is Sarah, and I am the biology lead for the DM1 program at Arrakis Therapeutics. So just a brief introduction of Arrakis Therapeutics. At Arrakis, or on planet Arrakis, as we like to say, we are developing small molecule drugs to treat human diseases. We are a platform-based company, and we have this DM1 program that lends itself pretty well to RNA-targeted drug discovery due to its nature of the toxic RNA foci in the cell nuclei. You probably don't need an introduction on the molecular mechanism of DM1, but basically the toxic RNA sequesters splicing factors such as MBNL1, leading to splicing dysfunction in the cell and subsequent cellular and functional symptoms. So for the DM1 program, we've developed multiple chemical series that selectively bind CUG repeats. We'll show you some data today on what our compounds can do. They bind CUG repeats. We've seen this through SPR and orthogonal methods. They displace MBNL1 in an in vitro assay. They disrupt the nuclear foci that contain MBNL1. We've seen a correction of splicing defects in both patient-derived cells and mouse model, and we've seen a reversal of myotonia in that animal model. So why small molecules for DM1? First of all, we think that an oral route of administration might be preferred by patients, but also small molecules lend themselves to a reasonably broad biodistribution. We've seen exposure in both skeletal muscle, heart, multiple other tissues, compound-dependent exposure in the CNS. And at this point, we are in late lead optimization. We're hoping to go into IND in the first half of 26. I just want to point out that our MedChem is structure-based right now. The structural biology team at our company has used some innovative designs for the CUG repeat RNA to be able to generate co-crystal structures with our small molecules that diffract reasonably well. So we have multiple compounds for which we've generated structures, and we'll show a cute little video here that shows how the small molecule threads through the UU mismatch of the CUGs. Okay, the first piece of cellular data that we like to show is the foci formation assay. We'll see if this works. So in this assay, what we've done is taken these patient-derived cells. This is an immortalized cell line from a patient with about 2,600 CUG repeats. We've taken these myoblasts, differentiated them into myocytes and myotubes, and then treated them with compound for 24 hours. After that treatment, we did staining. We stained via immunofluorescence for muscle-blind, or for the MBNL1 protein, which you can see here in green. We stained for the CUG repeats with FISH, which is shown in red, and you can see the co-localization in this DAPI stained nuclei in these foci shown in yellow. On the top row is our vehicle control treatment. And what I hope you can appreciate in the compound treatment is, first of all, a reduction in the number of co-localized foci in the nucleus, and also a broader distribution of that muscle blind in the nucleus. And for as far as the CUG repeats, we still see the same fish staining. We have a quantification on the right-hand side. What we have on the y-axis is the count of the number of co-localized RNA protein complexes per nucleus, and we've quantified several hundred per condition. In the gray is the vehicle control as the DMSO. The orange is 3 different compounds, and the blue is a negative control compound from the same series. This particular compound neither binds the CUG repeats nor displaces MBNL1 protein in our in vitro assay. You can see the corresponding data for that displacement assay at the bottom. So moving on from displacement to a functional readout, we next looked at the ability for the compounds to rescue splicing of MBNL1 targets. So again, we used those same immortalized cells, differentiated those myoblasts into myocytes and myotubes, and then we treated with our compound for 24 hours. Here we have 2 different compounds. Again, this 8164 tool compound, and then another compound here from the same series. We employed an amplicon, we isolated RNA and then employed an amplicon-seq based method to read out splicing for 6 different MBNL1 target genes. And then we use a composite score of rescue towards an unaffected control. And the y-axis, you can see the percent rescue towards that unaffected control. And on the x-axis, 3 different doses of that compound. We can see that an increasing dose gives an increasing amount of splicing rescue. We also wanted to make sure that this was not a unique effect on that screening cell line that we use. And so we took 2 more patient-derived cells. These are primary cells with different numbers of reported repeats, and we saw a similar effect. And finally, we like to show some of the in vivo data. We used the HSA-LR mouse model, in which about 250 CTG repeats are inserted off of a human skeletal actin gene, a transgene, in these mice, such that CUG repeats are expressed in skeletal muscle. And we first wanted to look at a pharmacodynamic readout of splicing. So we dosed our compound at 2 different doses. We have 10 mg per kg and 20 mg per kg IP daily for 5 days. And then we took down the mice on the 5th day, isolated RNA from the gastrocnemius tissue, and we looked at 5 different MBNL1 target genes and the amount of rescue towards the FVB background or wild type control amount of splicing. And so here this is not the composite score. We're just showing 3 different genes, but just for example, on LDB3, we see a rescue in a dose-dependent manner. Again, 10 mg per kg rescues about 30%, and the 20 mg per kg rescues about 60% of the splicing towards the wild type control. Furthermore, about 4 hours after the last dose, we did an efficacy readout. This is a muscle relaxation readout. In this mouse model, it's a plantar flexion readout where the mice are anesthetized, the calf muscle stimulated, and the force on a foot pedal is measured. And what we're looking at here is the time for relaxation. we can see that in the FVB background control, there's a fraction of a second in the time it takes to relax that muscle. In the HSA-LR vehicle control, it takes about a second to relax the muscle. And in the orange is the 5 days dosing of our compounds. And you can see the amount of rescue towards the wild type, again, in a dose-dependent manner. We also have an ASO control for this assay. This is an ASO that's directed towards the human skeletal actin, and that also works at 5 days. So this is the last piece of data. Right now, the focus of the group is to combine oral exposure with potency and also other drug-like properties. And we're continuing the march towards that goal. And we're really excited about this program. We're happy to talk with anybody after this. My colleague Emily, who is the lead chemist on the program, has a poster, and she's happy to talk with everyone too. And with that, we'll thank all the Arrakis employees, past and present, who have made this program possible. We'd also like to thank our KOLs, including Dr. Melissa Hale and Dr. Nick Johnson, as well as some of the other folks who have given us advice, Dr. Maurice Swanson, Dr. Charles Thornton, and Dr. Thomas Cooper gave us a plasmid, and our CROs, in particular, Myologica, who did a lot of our animal studies for us. So thank you, and we'll open for questions.
Thank you, and great talk. We'll be a little bit at devil's advocate here. So I'm just wondering, because you started your talk that this small molecule has the ability to penetrate the CNS, right? So what's the reason you use the HSA-LR mouse rather than the DSM XL? Because that would have been a great opportunity to have the one that has the CNS pathology.
Yeah, that's a good question. So mostly accessibility. The HSA-LR model is accessible via JAX, and the CRO Myologica has a lot of experience looking at the myotonia readout. We are working on accessing the DMS-XL model. So yeah, this way.
Over here. Hi. Very nice. So it's interesting that the foci don't go away, particularly because with it's not me. It's been shown
with morpholinos that block the binding of muscleblind, that leads to dissolution of the foci. So have you looked at the level of DMPK RNA? I mean, one thing you might think about is that it actually is starting to accumulate if this molecule is bound to it and the foci can't dissociate.
Yeah, so we have looked, I think we've looked about 3 days out, we've looked about 3 days out in cells, and we haven't seen differences in transcript across the board over multiple compounds. In the animal model, I think we've looked over about 5 days dosing, and we haven't seen any change in the DMPK transcript level. But yeah, we don't know past those time points if anything might happen. We also don't know if the compounds would stabilize the structure enough that it won't disappear. Yeah.
About the affinity of the compound for the CUG hairpins and whether the hairpins are stabilized. And I think from what you just said there, you don't stabilize the hairpins in terms of
Yeah, go ahead.
melting temperature or something like that?
We have looked at melting temperature, and I think again, like Emily knows more about that than I do. We don't consistently run that on every compound. We have evidence that it does. Okay. Thank you. And as far as affinity, I don't remember what our strongest affinity is, but it's I think in like the 100 nanomolar range.
So, another question, in terms of looking at the effect of alternative splicing in response to the compound, are you also treating the non-affected cells and wild type mice with the compound as well? Because sometimes compound splicing can be quite dynamic, and so small molecules can also affect splicing in normal cells as well.
Yeah, we did in the beginning when we were developing the cell-based assay. Since we use it for screening, we don't consistently look at that. As far as treating wild type mice, as far as I remember, we haven't done that yet. But that's an idea. Thank you.
Thank you very much. So now let's take a 10-minute break, but only 10 minutes, okay? Okay. So let's stick to our allotted time slot. So 5 minutes and 5 minutes Q&A. So that means we need questions, right? Questions are really important. They're important for the community and for the recording and all of those things. And remember, if you go over, you get later to your beers. So the first speaker is Dr. Haraldson from Avidity.
5 and 5. 5 and 5.
All right. Hello, everybody. Good afternoon. I'm Jeff Haraldson, Executive Director of Global Medical Affairs at Avidity, and super excited to be here to give you guys an update. I was told 7 and 3, so I'll truncate. Here we go. Which one goes forward? Big green button. Quick disclaimer, del-desiran, a lot of exciting data. Still an investigational product. This is clearly written by our legal colleagues. At Avidity, we're super committed to improving the lives of people with myotonic dystrophy and others. We have other neuromuscular programs in development with our AOC construct, and I'll share a little bit about that with you in a minute. Exciting RNA therapeutic. Everyone knows this. I think what I want to focus on here, considering time, based on prevalence papers and estimates, we know that maybe about 80,000 individuals with DM1 across the US and the EU. But the number that really is stark that I pay attention to is that blue number, and that's a 0. And we certainly hope to be the first product to market globally. Very under-recognized and progressive disease. And we are really paying attention to that with del-desiran, which targets the genetic cause of disease. You've heard a lot about the mechanism. I will not go into that in any great detail, but obviously a lot about the hairpin loops, and that mutant RNA is obviously very good at sequestering MBNL, which leads to a lot of MBNL-dependent pre-messenger RNA splicing downstream, which is believed to be a big cause of a lot of the manifestations that we see in these patients. Del-desiran is an antibody oligonucleotide conjugate, or an AOC. And here we have a DMPK specific siRNA, or a small interfering RNA that targets muscle very well, gets in and degrades DMPK messenger RNA through hydrolysis, reduces that expression of DMPK, and really frees MBNL back to its functional capacity, and hopefully corrects all that missplicing. In the MARINA, we've shared this data before, our phase 1, phase 2 trial, where we have shown here in the bars a dose-dependent actually increase in liberating free MBNL. Most important thing really is focusing on functional outcomes and improvement for these patients, these individuals with DM1. Video hand opening time, or VHOT, a really important indicator of targeting that chloride channel, for example, and looking at hand grip, QMT, lots of muscle strength and function outcomes in Marina, also in our phase 3 trial, which I will share with you as well. And not to skip out on the activities of daily living, which is extremely important in the assessments as well. So the AOCs, for those who are not that familiar with them, antibody oligonucleotide conjugates, a quick peek under the hood. What we basically did, the first company to do this, is essentially take two well-known and understood technologies, monoclonal antibodies and oligonucleotides, and link them together. Oligonucleotides are very good at targeting RNA. Since we know the sequence of messenger RNA, for example, on DM1 with DMPK, it just makes good rational sense that this would be a great a therapeutic to design. The Achilles heel of oligonucleotides or in the history is that they've done really well at targeting the liver and have had a hard time moving beyond that. So INWOX, our proprietary monoclonal antibody targeting transferrin 1 receptors, expressed throughout the body, but heavily expressed on skeletal muscle, cardiac muscle, smooth muscle, and that really shuttles the oligonucleotide to the site of action, into the muscle. We have been the first to demonstrate that in terms of really delivering big payloads of oligonucleotides across all of our neuromuscular programs, DM1, DMD44, as well as FSHD. Quick recap on Marina for those of you who are not familiar here. Marina phase 1, phase 2, again, with the primary objective of safety and tolerability, escalating from 1 to 2 to 4 milligram per kilogram doses in a placebo-controlled fashion. And then those patients, 38 patients enrolled, 37 of those eligible patients moved into the open label extension trial. And all of those 37 patients remain in that trial to this day. We did have one patient in the 4 milligram arm that was discontinued early due to an SAE. The safety and tolerability is really important. We're learning a lot about that. You can see we've had over 340 infusions of del-desiran, totaling 80 patient years of exposure. So that gives us a lot of content, a lot of data around. That fine? Wow. Are you going to cut me off? Here, let me go real quick. We know this, Harbor, phase 3, because this is the most important. That has informed Harbor, our phase 3 trial. So we have, the goal is enrolling 150 patients in a 1-to-1 randomization, del-desiran to placebo, 4 milligram per kilo every 8 weeks. And then they'll all get the opportunity to go into open label, just like in Marina. The bottom blue bar, really important. We are planning to complete enrollment in this phase 3 trial middle of this year. That's just a couple months away. And again, I think our primary endpoint, 30 weeks, looking at VHOT and opening time, and these other really important functional outcome measures as secondary endpoints. So that's it. Thanks, Andy.
Definitely very encouraging. And it's a focus on, but presumably it will target other tissues as well. Is that correct? Heart and perhaps the GI tract, the smooth muscle of the GI tract?
Correct. In our preclinical work in the NHP model, the siRNA was delivered most robustly to skeletal muscle, but also heart had almost similar DMPK knockdown. Gets into the diaphragm as well. So yes, it is pretty ubiquitous spread. It knocks down a little bit into the GI tract as well. So it definitely has other targets, but primarily it's been delivered to the skeletal and cardiac muscle.
Any thoughts on outcomes of what?
Yeah, sorry. Felt some. So any follow-ups? Patients start saying, "Hey, my GI symptoms aren't quite so bad," or something like that.
So
captured.
Anecdotally, we have heard from our investigators exactly those sorts of outcomes. Yes.
Great. Thank you very much.
Great. Thank you.
Next, we hear from where is Dr. Dugard and Dyne Therapeutics. The floor is yours.
Thank you. Hi there, everybody. So first of all, I just want to thank Tanya, Andy, Bas, Alain, MDF, and Euro-DyMA for giving us some time to talk to you today. Without them, we would not be where we are as a community. So thank you very much. Andy, Tanya said I had 30 minutes for this. Is that cool? There's a lot of slides here, 16 slides. I'm not going to go through all of them, but this is for you to have after the meeting. It's really important. I always like to start with this slide. At Dyne, we are there to deliver life-transforming therapies for patients living with neuromuscular diseases, with myotonic dystrophy type 1 being a priority. We have other programs, and we're trying to advance our platform for a number of disorders. We talked a lot about splicing today. Really, just the main message here that you'll hear me say a couple of times in these few minutes is splicing is the underlying pathobiology of myotonic dystrophy type 1. It occurs in a number of organs, systems, and leads to manifestations across all of the muscles that we've been talking about: smooth, skeletal, including diaphragm, cardiac, as well as CNS. So our clinical candidate, Dyne 101, addresses that central pathobiology to be able to correct the splicing, the missplicing, and lead to functional improvement. And I'll show a little bit of that data very quickly here. So we're able to correct the dysregulated splicing, and then we see early clinical effects with myotonia, but we've learned from all of you that myotonia is something that individuals learn to live with, but it's really the muscle weakness, the function, the CNS-related manifestations that really impact quality of life, the GI symptoms. And, our approach to myotonic dystrophy type 1 allows us to get into all of these compartments, including the CNS. The force conjugate does cross the blood-brain barrier, which is really important for these fatigue, sleepiness, cognition issues that occur as well. Very importantly, we have not done this in isolation. Many of the physician scientists here have given us input into our ACHIEVE phase 1/2 clinical study, as well as advocacy leaders that are sitting in the room, as well as patients and caregivers who are also experts in this disease, obviously. And so we thank you for giving us your input that has directly influenced the design of our phase 1/2 study. So this is our study. What I'll just really point out here is that we've gone through a number of steps to identify what we believe is the appropriate dose and dose regimen, which is 6.8 mg per kg, dosed every 8 weeks, and it's delivered systemically. And this is the ASO equivalent dose, just to point that out. We've completed the MAD portion, and we actually have all of our patients at the 6.8 mg per kg dose. Here you can see we're in 18 to 49-year-olds, and we're looking at safety and tolerability, as well as a number of other endpoints. And I'm pleased to say we've started our registrational cohort at the 6.8 mg per kg dose every 8 weeks. Overall, our safety profile has been very favorable from our perspective. Maybe a couple of things I'll point out: that we've had no drug-related SAEs, and that we are seeing some liver enzyme elevations in a minority of patients, but that's also due to the underlying disease. So we've talked a lot about target engagement, knocking down the DMPK. And here you can see 3 months after we've dosed with 6.8 mg per kg in the teal bar versus placebo, which is the gray bar, we're seeing a 33% knockdown of DMPK. So we're engaging the target in the muscle. And this is also leading to 25% improvement in splicing. Our preclinical data indicated if we can get to 20 to 25%, we should be able to show functional improvement. And indeed we are. Again, here is myotonia as measured by video hand opening time. And you can see in the teal line, we have about a 40% improvement from baseline in myotonia, and a big difference between the drug and placebo. We also looked at a number of other functional measures, such as QMT total, which is a measure of strength, 10-meter walk run, and 5 times sit to stand, which is particularly interesting because it's really lower limb function and dynamic balance. And we see an improvement out to 6 months with our 6.8 mg per kg cohort in all of these measures. So it's the totality of this functional improvement across multiple measures that's giving us so much confidence in moving forward with the 6.8 mg per kg dose every 8 weeks. And I'll just point out one quick thing. So Dr. Johnson talked about the natural history study that shows the correlation between splicing and function status. So at baseline splicing, depending on that level, you can see different levels of functional status at 3 months. And the Provenzano paper illustrates this beautifully. So splicing index is predictive of clinical function. And in our study, we're able to show that change in splicing at 3 months leads to functional improvement at 6 months. So we're able to correlate that change, that improvement in splicing from baseline to 3 months, to the change in function, the functional improvement at 6 months. And of course, the CNS, the MD high total here, we can see an improvement, about a 40% improvement on MD high. And if you look at a number of subscales that are CNS related, we're able to see sleepiness, fatigue, cognition, those very impactful quality of life symptoms improve as well. And again, we've shown in the past preclinical data that the force conjugate crosses the blood-brain barrier. So with that, I will take questions. So, yeah. We saw, yeah, exactly. We saw 20-25% infusion-related reactions. Many of them are sort of typical with what you'd see with IV drugs.
Like skin rash sort of thing.
That's right. Pruritus. Yep.
Exciting results, seeing this 25% splicing correction at 6.8 is really, really great news. I think I remember last year you talking about going up to higher doses before moving into the phase 3, and I saw that you had no SAEs. Can you comment about the decision to go in at 6.8 versus pushing higher?
Yeah, sure. So if you think about the data we've actually generated over the last couple of years, we've actually shown data from 1.8 mg per kg all the way to 6.8. The protocol allows us to go to 10.2 mg per kg ASO equivalent. And so what we're seeing is we're hitting, we're engaging the DMPK target the way we thought we would, the 30 to 35% engagement, knockdown of DMPK, leading to that 20 to 25% splicing correction. So we feel like we've actually gotten to a dose that has still a very wide therapeutic window, a safety window, and delivering the kind of efficacy that we want to see. So could we go up higher? Absolutely. But we do feel like we're kind of at that sweet spot with 6.8 mg per kg. Don't give the mic to a Luke. Please don't give him the mic.
Hi. It's interesting that, the MD high and the cognitive data has changed, and also, it kind of show on the preclinical data. Do you have other, evidence to show that, like, for example, but plasma biomarker or something that, it showed that it's getting to the brain?
Yeah, so I probably don't have time to go into all of it, but we have presented preclinical data. I presented preclinical data that shows a few things. So first, in the HTFR DMSXL mouse, right? So we have the DMSXL mouse that expresses the human transferrin receptor. So we've been able to show that we get into various compartments like the cerebellum and cortex and actually have foci reduction. And then we've also seen in NHPs, we've shown in NHPs that we get into not just the cortex with IV delivery, but also into the deep brain, the caudate and putamen. And we've compared this to intrathecal delivery of the ASO, as well as just naked ASO. And so intrathecal gets into the cortex, but not deep brain. And IV unconjugated doesn't get into the brain at all. So we've shown a lot of preclinical data that gives us that backs up kind of what we might be seeing here in the clinic.
Okay, this is a very easy question for you this time.
There's no easy question with you. but
yeah, more focused question.
My time's up.
Oh yeah. So do you see a liver toxicity, because you mentioned that there is elevated level of liver enzymes? So do you see liver toxicity?
We don't see any liver toxicity. We don't see any. So we've looked at, for example, bilirubin conjugated, unconjugated. We see no liver dysfunction, no liver toxicity at that level. But again, we see liver enzyme elevations, but it's also confounded by the baseline liver enzymes that occur in the natural history that Professor Sansone and others have shown. And there's a lot of PIs here too, so you're welcome to ask them to kind of what their experience is. I would encourage you to do that.
The next one presenting is Dr. Jane Lockendale from PepGen. Take it away.
Thanks, Andy. So to move this down, I'm trying to cut it short. Thank you to the MDF for allowing us to speak today. Like everyone else, I probably have too many slides, so we'll go quickly. Disclosures. We're a company that is committed to transforming the lives of people with myotonic dystrophy and other neuromuscular disorders, and we're developing our enhanced delivery oligonucleotide platform. Our enhanced delivery oligonucleotides are PPMOs, peptide-linked PMO oligonucleotides, which are designed to get oligonucleotides into the nucleus. I don't need to belabor the fact that delivery is important for oligonucleotides, but I think you can see beautifully here on this slide that untreated cells have no drug, no surprise there. Treat them with a naked PMO, very little gets into the nucleus, but you can see a lot of red once treated with our PPMO, which gets the oligonucleotide into the nucleus very effectively. We are an oligo that's designed to bind to the repeat sequence, so we target only the pathogenic DMPK. We do not knock down DMPK. We have non-clinical data showing we deliver to a lot of different systems that are affected by myotonic dystrophy. Very quickly, non-clinically in cells, you can see here that we reduce the number of foci. We liberate muscleblind, which is the goal of our therapy, and we indeed correct splicing in a dose-dependent manner in cells. Similarly in mice, we get the drug in, more doses gets more drug in, not surprisingly. More doses gives us more splicing correction over time, and more doses give us better correction of myotonia in the HSALR mouse model. So this and a whole lot of other data, which I'm not doing my non-clinical colleagues any justice by not sharing, led us to start our clinical program. We currently have 2 clinical trials ongoing, Freedom, which I'm going to share some data with from here, our single ascending dose study. We have dosed our first 2 cohorts, 5 and 10 mg per kg. We're currently dosing at 15 mg per kg. And this is a single ascending dose study. So I'm showing data from just 1 dose. Importantly, in terms of demographics, our groups are reasonably well balanced. I will point out the 10 mg per kg had a slightly lower splicing index. That means they were less bad, if you will. They were closer to wild type and not as close to a severely affected person with myotonic dystrophy. And importantly, and we'll come back to this, our VHOT in our placebo group was 14, which means they were very severely myotonic in that placebo group. And you will see in a moment why that becomes important. Safety. Any single ascending dose study is, of course, a safety study. We did have some treatment emergent adverse events that were mostly mild or moderate, nothing too significant, but we did have 1 severe adverse event in the 10 mg per kg group. Unfortunately, this participant took an off-label drug the morning of our infusion. This rather confounds the use. Her PI didn't know about it. We didn't know about it. And she had some severe abdominal pain, which resolved very quickly. So we did have a severe adverse event, but it's a little bit confounded. We had a couple of serious adverse events that were not related to the drug as well. One was related to the biopsy procedure and resulted in a lot of retraining for our PIs. One of the things you worry about with oligonucleotides is the kidney. A lot of oligonucleotides affect the kidney. This is just showing that effectively we had no effect on creatinine at all in these 2 dose levels. Efficacy. Well, we got the drug in. It's dose dependent. We get more in at 10 mg per kg than 5 mg per kg. That is always a good thing to see. And to be honest, we were surprised by this. So these 2 low doses, we saw very much dose-dependent increases in splicing. 5 mg per kg, 5 out of 6 people saw improvements in splicing. 10 mg per kg, 3 out of 4 did. We had an issue with one of the samples. And this increase in splicing was dose-dependent. I have to say we saw the 5 mg per kg data and said, looks good, but we don't really believe it. Then we saw the 10 mg per kg data, and now we're really excited to see 15. These are really nice amounts of splicing to be seen after a single dose. Functional outcomes were perhaps not as encouraging. We really didn't expect to see much functional change after a single dose. On the left, you can see the 10-meter walk run. There was a slight improvement at the 10 mg per kg cohort, but we're not really convinced yet. Myotonia? Well, everyone improved, but I think you'll see the placebo improved more than anybody else. Our placebo is really good at correcting myotonia. I mentioned at the beginning that that group had very severe myotonia coming into the study, and we believe that this is just baseline variance and variance in myotonia regression to the mean, if you will, in these people with very, very high myotonia coming in. And that is a problem with small studies. So with that, to conclude, we have a treatment that targets only the pathogenic DMPK. We have a favorable emerging safety profile. We have dose-dependent increases in tissue concentration across these first 2 cohorts, and really nice dose-dependent improvements in splicing across these first 2 cohorts. given this is early days with the single ascending dose study, we're very encouraged by where we are. We have an ongoing multiple ascending dose study as well that I'm out of time, so I won't go into in detail. But as we're dosing the first cohort, we have no data to show anyway. But with that, I want to thank everybody who's been involved. I think Ash said it very well. It takes a community to build a drug program like this. We've spent a lot of time talking to a lot of people in this room, getting your advice, talking to people with myotonic dystrophy, talking to their families, talking to advocacy groups, PIs, all our many, many collaborators. So thank you all very much. And I'm very happy to take questions.
Well, going once. Okay, no question. Well, thank you so much, Jane.
So the next presenter is Dr. Alain Weidenfelder from Lupin Neurosciences.
Thank you. Thanks, Andy. My name is Alain Weidenfelder, and have been heading the medical affairs at Global Specialty at Lupin Neurosciences and serve as medical lead on our clinical program. Actually, Andy, I do have an email from you that tells me that I have 10 minutes. So don't set that for 5. All right. You made me nervous. First of all, a disclaimer that all the information that I'm going to present is solely for an educational purpose and is not intended to promote the use of our products. Namusca has not been approved for pediatric use, and I'm going to give you an update on our mixilitin program for myotonia. So we're getting more close to the clinical stage of exciting programs that we have seen over this afternoon from our colleagues in the preclinical stage. And I'm going to give you some more insights on the clinical part, and in particular, talk a little bit about the myotonia. So the myotonia is the inability to relax a muscle after voluntary muscle contraction or muscle percussion. And I'm going to make you see and also hear the myotonia because I have a couple of videos. They should work, but I don't know how to point the cursor. So just click. Yes, it's working.
Serrez la main. Fort, 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 fort. Ouvrez vite.
The colleague recognize the voice of the physician. So myotonia, they have the common feature across several myotonic disorders, yet they are separate from muscle myopathies. And this is important because with myotonia as a single symptom, there are other comorbidities that come with that, and that is fatigue, it's the pain. We're going to touch base a little bit on the impact that myotonia causes on the quality of life of the patients. But first of all, so as a spectrum within the muscle disorders, the myotonic disorders fall under the muscle channelopathies, and the one that we're going to talk about here a little bit more is actually the non-dystrophic myotonias. So they're separate from the DMs, as we know, and they have different pathologies based on the gene being affected. So it's the chloride or sodium channelopathy, and the complexity around this is the fact that depending on the gain or loss of function of the chloride one, we have a different phenotype of the disease, although there is the same symptom. So the triggers that are provoked by underlying muscle channelopathies would differ strongly among these different subtypes. And it is important to know and to recognize in order to accurately diagnose the type of the myotonia, but also to adjust the clinical management. So it's not a diagnosis, myotonia, it's a symptom. One of the symptoms for DM1 causing the or one of the symptoms of the DM1 among the other systemic complications that we've already been talking here about, but it's the only symptom for the NDM. These people are otherwise quite healthy without not too many comorbidities, and that's what it makes really important in terms of the unmet need and the urgency to handle myotonia in these patients. So the pathogenic differences mean that the systemic reach of NDM is also different. And we kind of also had to take that into account in our pediatric program as well. Diagnosing suspected myotonia is the combination of detecting the clinical muscle stiffness combined with the family history, but also the exercise. So here's where I'm going to make you hear the myotonia with the help of the EMG. Very good sound here. So what makes it really complex, especially for the muscle channelopathies, is that in the absence of the family history, because there are several subtypes that are of recessive inheritance, it makes it more difficult to diagnose these patients. So the role of the genetic testing is extremely important. However, the EMG and the knowledge about the exercise type of the tests is also helping to expedite the diagnosis. Before we go into the results of the clinical program, just wanted to talk a little bit about the clinical manifestations, but I'm afraid of Andy's clock. So I guess the only thing that I wanted to emphasize here for the congenital DM and childhood DM, because again, it's the pediatric program, that usually myotonia manifests a little bit later, and it certainly precedes the muscle weakness and wasting. Yet it's important to understand what impact it may have on children. So we've gathered some insights both from clinicians during our advisory boards, and we conducted some surveys, but also scientific literature, which does show how complex the picture is for the NDM patient journey, with the hurdles, barriers, delayed diagnosis, and different milestones that the patients go, especially at the time of their development. And this all is important also to take into account when you're taking patients into clinical program in therapeutic development programs. And I have to say that we've learned a lot about this patient group conducting this pediatric investigation program. So the whole program was about safety and also understanding the pathology of mixillotin in younger patients. So we have taken it in a staggered way. So the patients were enrolled subsequently, starting with the older cohort, the young children of 12 up to adolescence age. And based on the safety data and the PK, so we've developed a model, PK-PD-POP model, to inform the exposure, and also based on the safety outcomes, the second cohort has been enrolled. It's a very short study with only about 2.5 months. Patients were only 4 weeks on actively on the maintained dose. They were all offered to go into the open label extension. They were followed up for 2 years. So it is a study which had a lot of clinical visits. Again, I've emphasized that these patients do not have a lot of comorbidities. They're scholarized. These are all points that need to be taken into account when you conduct the clinical trials. We do have a registry also for the patients younger than 6 years old. And we look into the mixillotin use in patients from 0, so from newborns to up to 6 years old. Some demographics. So we had about seven patients in the first cohort and 5 patients in the second one, with a good distribution of gender and a good representation of different types of the myotonia or channelopathies, as well as DM1. The mean age across the cohorts, and the body weight. The body weight is important, and also the BMI, because it will inform the mixillotin exposure and the choice of the treating dose for this patient. So the most common ARAs were the abdominal pain, nausea, and nasopharyngitis and headache. And there was not more than 10% of patients that were affected by adverse events. I think the most interesting as well, and I do have a more detailed slide on the safety. It's not, of course, not that, it looked a little bit maybe simplified. But I think it's also exciting to talk about the efficacy endpoints. And some of us came from the Euro-CRN meeting that was organized by Professor Rosa. And the topic that we really discussed and dwelled on a little bit is the development of the clinical endpoints for patients with myotonic dystrophy. And it is challenging because a lot of them are not fully validated. And when it comes down to the pediatric patient population, it makes it even more complicated because a lot of this is also something that we look into patient-reported outcomes, and children, they are not small adults. They also would see things differently from the caregivers, and also the subjectivity of the surveys and the questionnaires that we would give to pediatric patients is a question of what do we need to do to validate all these questionnaires and the surveys and the way how we measure the therapeutic impact. So we went with the visual analog scale for stiffness. We also have data for pain and fatigue, but also the myotonia behavior scale. And of course, this is because we were interested in myotonia and myotonia improvement. The MBS goes from 0 to 5, with 5 being really impacted or having a severe myotonia and claiming that it has a significant impact on the quality of life. And 0 to 1, that only says either I don't have the myotonia, or I have some myotonia, but it doesn't impact my life. So you can see that most of the patients, some of them didn't have any change based on the MBS. Most of the patients, they did have an improvement on the MBS scale. And this is quite encouraging because this is the first time that the MBS has been tested in pediatric patient population. What is even more interesting is that we could show that there is a strong correlation between the PET quals of children and parents. So, and this is at the baseline. There was a really significant improvement of the PET quals from children, and also responded by the parents on the right-hand side, for in particular the physical and total scores. For the emotional one, only reported by children. Another interesting measure, and I think you may also be interested in this one, is the hand grip relaxation time. We use the dynamometer, the QMA system, there is a weak correlation between the IGRT and PET score, but that just also tells us that, again, something that leads to the improvement of myotonia also at this very young pediatric age means something in terms of the improvement of the quality of life of people. You can see that the changes from baseline to end of study, we don't have any stars here, so the p-value was by 0.05, but it's only a few patients, so we're talking here about 10 patients, yet there was a strong trend towards improved hand grip relaxation time. Last but not least, I just added some combined cohort data for other domains of the PET quals, both from children and from parents. There is a trend on improvement, but of course, symptomatic treatment, again, as I've said, it's the only symptom that these patients have, yet it does have an important impact on their daily life, and mexiletine can indeed really have some positive benefit in a very short period of time, 2.5 months, so 56 days of treatment. This one is also interesting. It just shows the exposure, and no matter of the type of the diagnosis, gives you the daily dose of mexiletine, and just demonstrates that they were all in the therapeutic range. That's it. So the conclusions are that all these children were enrolled in the long-term follow-up. up to 80% of physicians said that the mixed treatment was either good or very efficient. And across the physical and quality of life measures, so trends towards improvement were also observed. Thank you.
I apologize if I missed it, but did you check maxillatine levels in the blood?
Yes, it was in the last.
Oh, okay.
Sorry,
I missed that.
Yes, we looked at the, I mean, of course, the values, they are all corrected to the body weight.
Okay.
But we also know that there is a dose-dependent increase as you escalate. But all the patients were also in the therapeutic range, which is between 0.5 and 2 milligram per ml.
That's really good because, as you may know, in the adult study, the levels dropped down, and the question was, were they not taking it, or is there some metabolic increase that gets metabolized quicker? But so that's interesting.
Yeah,
Thanks.
there are some metabolic impact, like for example, smoking or other things as well. Sure, but the compliance in these patients is also a question. Yes. And indeed, we, as you've seen, we did have the DM1 patients in the study, and it was indeed a little bit more difficult to keep DM patients compared to the channelopathies on treatment at this age in particular. Yeah. All right. Thank you. We can chat.
Thank you very much. So before we get to the last presentation, a quick housekeeping request. Can all the presenters who presented in this Pharma Day session afterwards just come to the front, because we need to take a photo for our friends from Euro-DyMA. And now, last but not least, Dr. Walker and updates from ARTHEx. Thank you.
Thanks, Andy, and thank you to MDF and Euro-DyMA for inviting us here to talk about our program. ARTHEx Biotech is a small biotech company based in Valencia, Spain, and we are developing a drug for myotonic dystrophy, initially DM1, but hopefully also DM2 eventually. So we are a clinical stage company. We are in the clinic at the moment, and I'll tell you a little bit about our ongoing ARTHEMIR trial, which is in adults with DM1. What's interesting and differentiates us a little bit is that we have a dual mechanism of action. I'll talk briefly about that, but there's much more information at our posters. I've got my colleague Diego here so please reach out to Diego or myself after the talk or anytime during the next few days. Not much I can say in 5 minutes, but we'll try to do our best to convey what we're doing. We are also going to be looking at pediatric DM1 as well in the future. So let's talk about our lead program, which is ATX01 for myotonic dystrophy type 1. Of course, you all know about myotonic dystrophy, but one of the things that has been highlighted by others and that I also want to emphasize is that this is a disease, of course, of muscle, but not just muscle. And so our drug aims to deal with a lot of the different aspects, including muscle, but also outside of the muscle, and in particular, the brain. Next slide. So one of the things that makes our drug different is the target. So our target is a microRNA that's very much involved in the pathophysiology of DM1. That's microRNA 23B. And that leads to a dual therapeutic approach. When you inhibit or block miR23B, you decrease DMPK and you release MBNL, but also you increase the production of MBNL. And I don't have time to go through exactly how that happens, but we have a poster that talks about our mechanism of action. Also, what differentiates us is our delivery method. So you've heard about different delivery methods. What we do is we link our oligonucleotide to oleic acid. Oleic acid is actively transported in specific tissue cells, including muscle, heart, diaphragm, and CNS. So we are currently running the ARTHEMIR trial. And this is a first in-human study in patients, adult patients with DM1. We are running the study in the US, Canada, the UK, and Europe.
As any first-in-human study, our primary goal is safety and tolerability, but we are also looking at pharmacodynamic biomarkers, including splicing and MBNL levels, and we're looking at exploratory clinical endpoints such as the usual VHOT, 10-meter walk-run test, etc., as well as patient-reported outcomes. So we have two parts to the study. Again, you're hearing the same thing from many different sponsors. We have a SAD, single ascending dose part, and a MAD, multiple ascending dose part. In the single ascending dose part, we have 4 dose cohorts, and I'm happy to say that we have completed the first dose level. Each cohort has 8 patients, 6 on active and 2 on placebo. This is randomized. And we are starting with dose level 2. We're going to be doing muscle biopsies at baseline and either 15 or 28 days later. We are following patients for 3 months after the single dose. And the multiple ascending dose, we have 3 dose levels, starting with dose level 2 that was used in the SAD, and we are giving 3 doses every 2 weeks. We do expect, based on our animal data, that we will be able to dose less frequently than every 2 weeks, but to ensure that we hit proof of concept, we are dosing every 2 weeks in this study for 3 doses. And again, we're doing muscle biopsies as well as looking at clinical outcome measures. The interesting thing is because we target, we increase MBNL protein independently of the gene defect, we could also have potential for DM2. And as you know, DM2 has a different presentation, different symptoms, but we are now working with many of you in this room at developing our program for DM2 and hope to get that going in the near future. So we're quite ambitious. We're a small company, but we're very ambitious. So in addition to doing our DM1 study, which is in clinic, and our DM2 program, which we're also planning to look at congenital DM1. We have also some other compounds that are focused on other muscular conditions as well as neurological conditions. And on that note, I'd like to thank you for your attention. I know it's been a long afternoon. I'd like to thank the patients in particular who we've learned a lot from over the course of the last few years, and look forward to having many more discussions with you. Thank you.
Well, if there are no questions, thank you very much.
Thank you.
Yep.
So as Tanya mentioned, there's a reception now, right? The pre-conference reception, I think it's upstairs. So please join us there. And those who presented again, please come up front for a photo. Thank you.